Okay, so we're going to look at part two of our breakdown catabolism of amino acids, in particular the carbon groups. What I want to say, first of all, is we're not going to get through all of the amino acids. Some are super complicated and require full lectures on their own, but they're very interesting transformations. I'm just trying to hopefully get us a little bit of a baseline so we can see how a lot of this stuff is just all interconnected, right? We want to do this catabolism and this breakdown. We could just excrete these things if we didn't need it, but we can catabolize these things, break them down, use them in other processes that are productive in biology. Okay, so what I want to focus on to start is the catabolism of arginine. So arginine has the guanidino group attached to it. We actually talked about how to make this amino acid. And so it's often the case that one of these nitrogens is protonated. It's just that basic of an amino acid side chain. We have the NH2 up, and we'll just pick on the neutral form of the carboxylic acid. Realize it's probably not the best practice. And in fact, let's go ahead and just do the good practice. Okay, NH3 plus. There. Everything's got a happy protonation state. Okay, so what we should recognize right off the bat is that it's possible for arginine to be hydrolyzed with water, a process that occurs in the urea cycle, and that's going to actually form urea as well as this NH2 bearing amino acid. And this is ornithine, which we've talked about before, so we spit out urea. I'm surprised the book doesn't mention that this is an enzyme catalyzed event. Seems like it should be. Arginine being that stable in aqueous solution, seems like an enzyme should promote this hydrolysis reaction. But anyway, we're at ornithine. What can we do with that? So ornithine has an NH2 group at the end of the chain, and it turns out, and we'll see this again, we can react this in a transamination reaction with alpha ketoglutarate and in a transamination reaction through a PLP-dependent enzyme to swap places between that nitrogen at the end of the chain, that NH2, and the carbonyl of alpha ketoglutarate. Now what that gives us is a super cool aldehyde okay, a super cool aldehyde called glutarate semialdehyde. Now what we can do with the glutarate semialdehyde is simply oxidize it to glutamate. So that's going to require NAD+, capital AD+, to do an oxidation reaction and convert that aldehyde at the end of our chain into an acid, and then we've got our glutarate. Did I do that right? 1, 2, 3, yeah. Excuse me, glutamate. Or L-glutamic acid. Okay, so that's another amino acid. We've talked about transaminating that to get to alpha ketoglutarate, which is a very versatile metabolite. We can also take that semialdehyde and do something else with it. I'm just going to touch on quickly the biosynthesis of proline. So proline is a funky amino acid. It has the usual components of an amino acid, except the amine is baked into a 1, 2, 3, 4, 5. Goodness me, 5-membered ring, not a 6-membered ring. 5-membered ring. This is just such a fun amino acid. It's actually a very versatile catalyst, so I really like it. I think I've mentioned that a few times, or maybe I mentioned it in organic too. The whole world's blurring together in terms of my lecture content. Okay, so we have, we probably have 2 hydrogens there to give us an N+. Okay, so we can do the biosynthesis of proline using our glutamate semialdehyde. So this is my glutamate semi-aldehyde that was produced above. Now what I want to recognize here is that the nitrogen is 1, 2, 3, 4, 5 carbons away, 5 atoms away from the carbonyl carbon. So what I'm going to do is do an intramolecular imine formation, where the nitrogen engages the carbonyl to make an imine. Okay, and then all we have to do is take that imine, which has, if we count the atoms, 1, 2, 3, 4, 5, and we lost H2O as a result of that imine formation. And we can react that with a reducing agent, NADH, to give rise to NAD+. So we can react that with a reducing agent to form the corresponding amine. Okay, we probably need to add H+ to this. But the hydrogen goes here, and that's the H from our NADH that serves to reduce that imine. Oh, I had the wrong position. That's the alpha position. What am I thinking? What am I thinking? That H is going to go here. Sorry about that. Then we can add an H+ just to get back to the zwitterionic form. Okay, so pretty cool intermediate to give rise to another amino acid. So that's the glutamate semi-aldehyde. Now, bearing that transformation in mind, we can see how proline is metabolized. So proline, if you've got proline and you want to deal with it, it's pretty quick to see. Catabolism of L-proline is the opposite of its biosynthesis. So what we can do is we can take our L-proline and we can do a selective oxidation with NAD+ to give rise to NADH, and that's going to give rise to the imine that we just dealt with. And then that can react with H2O to give rise to the semi-aldehyde of glutamate. So this is the glutamate. I don't know where the semi nomenclature comes from. The glutamate semi-aldehyde, which can then be oxidized with NAD+ to give NADH. We just saw that. It's in the notes just right above where I'm writing here. I'm going to write it again, though. Form the dicarboxylic acid as the carboxylate. And that's just L-glutamate, as I just mentioned. That can engage numerous cycles to give useful metabolites. Okay, so there's proline, there's arginine, there's the biosynthesis of proline. We're really hammering through. What I think is a nice one to end with is, the book does a nice job of breaking this down, is the catabolism of valine and how it relates to isoleucine and leucine. Okay, so valine's got like a V side chain, that isopropyl group. Isoleucine is a chiral molecule at the beta position. Within this depiction, we see a dash of the methyl group that's at that position, and it's an isomer of leucine, which has an isobutyl tail as opposed to an isopropyl tail. We don't have a second chiral center on this molecule. Okay, what we want to do in all of these is a transamination. transamination to convert the alpha center into a carbonyl. Okay. And then we're going to lose CO2. We're going to lose CO2 in every case and add CoAsh, or the thioester. We're going to add CoAsh to make the thioester. Now, CoAsh is it's the CoA thiol. That is the sulfur atom of CoA. It's not engaged in acyl group yet, so it's not a thioester yet. Okay, now that's going to convert all of these into the corresponding carboxylic, or excuse me, the corresponding thioester. Where instead of having a CO2, which was lost during decarboxylation in every case, we're now going to have a thioester. And this uses processes similar to what we've seen in the past. We can do kind of I think it's a TPP-induced decarboxylation event. I'd have to check more carefully. Yep, it is a TPP. So we could put in here TPP pathway to lose CO2 and then add to the lipoamide and then displace with CoAsh to make the thioester. So anyway, we're at the thioester now. And what we have here are some interesting intermediates. And it turns out that I think, let's see if the book walks us through this. This isobutyl, so these are each going to kind of go on to different things, but it looks like in every case we do a dehydrogenation. So we lose H2 in every case to make a double bond added to the system that's in conjugation with the carbonyl. So we're going to now have an alpha beta unsaturated carbonyl. I'm just going to rotate this as well, no, I won't. That's not good practice. That's not good teaching. Okay, so we are going to have a double bond here, S-CoA, then we're going to have a double bond here, destroying the other stereocenter, S-CoA, and then we're going to have a double bond here, S-CoA. So we get to these metabolites. Then what I want to show on the next few pages is that we can take these to things that can enter into the citric acid cycle. So with a few steps, we can get to succinyl-CoA. Succinyl-CoA. From here, we can get to succinyl-CoA also, in addition to acetyl-CoA. And then down here, we actually get to 3 acetyl-CoAs. Okay, so the next bit of this lecture, I want to try to go through the steps required to get to those useful metabolites. So we've done the steps shown here. We've done the transamination. We've treated with a TPP, a TPP-dependent enzyme to convert an alpha keto acid into a thioester of CoA. And then we've done a fatty acid dehydrogenase-like enzyme where we remove a beta and a gamma hydrogen to make a double bond that's called a desaturating enzyme, as it introduces the double bond. And then we're going to show how we convert those double bond containing species into the various metabolites that can be directly used within the citric acid cycle. So what I want to start with is the first intermediate. So I'm just going to redraw this carbonyl up, but this would be this species. Okay, so what we're going to do is add water to this intermediate and let it do a conjugate addition. That is, assuming we have a ton of enzyme involvement here, we can add to the beta position with hydrogen transfer. Water's not a great nucleophile on its own. That's why we have the enzymes to help us out. And then I'm going to allow the hydrogen to transfer into the enol alpha position. So that hydrogen was right here. The arrows that I showed directly actually make an enolate, which is rapidly protonated under aqueous conditions to give an H+. And now it's this species that undergoes hydrolysis with water at the thioester. So there's an arrow to get us started. We can come back down and kick out S-CoA. But that's going to convert us into a carboxylic acid. Okay. This species will undergo an oxidation reaction twice. So 2 oxidation reactions. That's going to require 2 NAD pluses to give us this methylmalonic acid. And the methylmalonic acid, I'm going to draw this hydrogen explicitly, will rapidly undergo decarboxylation to give this species, which can pick up the hydrogen that was just lost from the other carboxylate. It's probably an intramolecular reaction. So we have propanoic acid, which will react with CoAsh. It's got to be an ATP-dependent enzyme, I would think, although I don't see the set here. Okay, well, maybe they don't require the ATP. Maybe this maybe the malonic acid isn't involved like I thought it was. Okay, so it's probably likely that you go directly from here to here through the aldehyde intermediate. That probably allows you to avoid going through the malonic acid, which maybe isn't as prone to decarboxylation. I'm not so sure. I'm going to leave these up. Somebody maybe on the internet is mad at me. Maybe some students are mad at me for not having that clearly put out there. But, you know, I just think, okay, well, let's move on to the next intermediate. The next intermediate is fun because it has a retroclason reaction. So now we're going to start with a different alpha beta unsaturated thioester. We're going to start with this one. Just zoom out ever so briefly. That came from the top middle intermediate, which ultimately derived from isoleucine. We're first going to add water to this. And that water is going to go to the beta carbon. And we could show H plus adding to the alpha carbon if we need to. It's probably an addition, forming some sort of stabilized enolate intermediate, which then gets protonated. That's going to give this species after the hydrogen drops off of the OH2. That beta hydroxy compound is going to get oxidized to the carbonyl. And this keto group can undergo a retroclason reaction with the sulfur atom in CoAsh. So we can add to this carbonyl, come up, come down, go over to make an enolate on that 3-carbon chain. And we've got acetyl-CoA along with, I just realized, sorry, I got so distracted last time. Negative charge here gets rapidly protonated. I got to this intermediate and didn't take it forward. Okay, so we got to take this intermediate forward onto something, and I'm not going to show it here. I think we've already looked at this. But this intermediate is propionyl-CoA, and it readily forms succinyl-CoA. So I'm kind of cheating by not showing all of the steps. If you picture the dicarboxylic acid, so instead of CO2 minus, we have CO2H, and instead of a thioester on the right side, we have CO2H. That's a dicarboxylic acid with 4 carbons. O is 2, mi for malonic, S for succinic acid. 4 carbons is succinic acid. If one of them is the thioester of CoA, we call it succinyl-CoA. Okay. So we have succinyl-CoA produced along with acetyl-CoA. Now, what about this last intermediate that was derived from the isobutyl-containing leucine? Well, we are going to add a few things to this in the first step. We're going to continue our trend of adding water, but we're going to do so in an oxidative event at the gamma position. So we're going to do gamma. It's not oxidation, it's gamma CO2 addition. This is going to be done with ATP, H2O, and HCO3 minus, which HCO3 minus is just CO2 plus H2O, and then we lose a proton. Okay, all of this is going to put a CH2 at the gamma position. It's a very slick reaction as a carboxylate. Then we're going to add water in a Michael reaction. I'm just going to pick up an H plus from somewhere. So we're going to add the water to the beta position by way of a Michael reaction. That's going to put CO2 minus at the gamma position. The beta position is going to have an OH. We have a methyl group also at that beta position. Then we have this species. Okay, well, this species can undergo a retroaldol reaction. To give, so retroaldol, where I'm losing the hydrogen on the OH, to give acetoacetate, which is directly used in the citric acid cycle to make 2 times acetyl-CoA plus, S-CoA with a negative charge at the alpha position, which just picks this up to make the neutral species, which is simply acetyl-CoA. And we did it. Okay. So that'll do it for the catabolism. Again, there's a lot of great ones. The next one is the methionine catabolism is great because it shows how we form homocysteine and how we have to deal with that. Homocysteine can actually cause disease, and so your inability to fully break down methionine can be a huge problem. And then breaking down the aromatic rings of our aromatic amino acid side chains, very complicated mechanisms warranting full lectures in and of themselves. But for now, I think this was a good start. Hopefully it was clear enough. I know these structures get complicated. They do get easier with practice, so I can attest to that. But anyway, that'll do it for the catabolism. That'll do it for this little group of lectures. Next, we're going to look at the biosynthesis of amino acids and hopefully some mechanisms for the formation of polypeptide chains. Okay, and until then, we'll see you next time.